Hello everyone, welcome back to Big Orange Biology. It's Mr. Jennings here, and today we're going to talk about glycolysis and cellular respiration in a little bit more detail. We care about electrons and this whole unit. We've been spending time talking about this in class. We talked about these concepts called electron carriers. And now this information, the whole big thing about glycolysis and the Krebs cycle of cellular respiration is that the main thing they're trying to do is move these electrons out of high energy molecules like glucose and try to put them into something a little bit more usable, trying to take the chemical energy in glucose and harness into more easy to digest energy for cells. So how does this work is basically we want to move a lot of electrons. We're going to do it through various redox reactions, and the best way to move an electron is to often use hydrogen. So we're going to see this a lot, especially here in cellular respiration, but not just limited there. We're also going to see this quite a bit with photosynthesis. So the movement of hydrogen is also going to cause the movement of electron, and because they go hand in hand, because hydrogen's positive charge, when it's a hydrogen ion has a positive charge, well, the electron is going to be attracted to it. So cellular respiration of itself has four main stages that we like to deal with. First off, we have glycolysis, which is the main process that all life forms do for their anaerobic respiration process. It takes no oxygen. It occurs in the cytoplasm, or the cytosol, as you see here on the screen. Then you transition to the aerobic process, which includes things like the pyruvate oxidation, also known as the link reaction, the Krebs cycle, which is what we're going to speak about today as well, and ultimately the ETC, or also known as oxidative phosphorylation. All these processes are very valuable to the overall cellular respiration process. Now, ultimately, cellular respiration's main goal is to produce a lot of energy, and the biggest thing is using the enzyme known as ATP synthase. So keep this as an end goal in mind. All these molecules that we're trying to make are hopefully going to help power ATP synthase later in oxidative phosphorylation. Because what we're going to do is we're concerned with the flow of hydrogen ions. So we need to create a situation where hydrogen ions are flowing along with electrons. So we need to do things like creating a hydrogen ion gradient, what we call a proton gradient, and that's going to be a complicated issue, and it's going to be something that we're going to talk about in a future video. But keep in mind, all these processes are literally going to build up to the big event, which is ATP synthase and oxidative phosphorylation, which is a part of the electron transport chain. So glycolysis is the main start of this, because first thing we're going to see in glycolysis is we're going to take glucose and we're going to convert it into a more usable sugar called pyruvate. It's a 3-carbon sugar. Basically, pyruvate is a little bit more energetic to yield because it's got a little bit more carbon to strip off, and otherwise it can be easily oxidized. And if oxygen is available, pyruvate will enter the mitochondria, where a lot more action can take place. Enzyme of the Krebs cycle is going to help completely oxidize glucose from pyruvate into a CO2 molecule, which is going to help get that ratio of carbon to oxygen down to a 1:2 ratio from a 1:1 ratio. Now, overall, though, glycolysis is a 10-step reaction. We're not going to get into too much detail here, but the big thing is, one, you produce pyruvate. That's like the critical thing here. That's essential for Krebs cycle. But also, you're going to end up producing about 4 ATP and 2 NADH molecules. Now, unfortunately, to do this, though, it consumes a couple ATP molecules. So in reality, all you're making is about 2 ATP and 2 NADHs. That NADH right here, that's the electron carrier. It not only is going to carry that electron, it's going to carry that hydrogen ion, that essential proton, and that's going to help drive that ATP synthase later on. Now, cellular respiration of itself here is, again, we're right here in glycolysis. We've gone from glucose to pyruvate. Now we're going to go into the mitochondria. Now, going into the mitochondria in itself, there's a series of reactions take place. There's going to be the link reaction, which we're going to discuss briefly, and then the Krebs cycle. Just a little bit of facts about the mitochondria. We discussed this a little bit in the class, but we want to make sure I have here. So the mitochondria is a double-membraned organelle. It's used for energy harvesting. It's got that smooth outer membrane. It's highly folded on the inside. Those folds are known as the cristae, and it also has an intramembrane space. So the folds are separated, and that's what we see here. There's actually a fluid-filled area in between the membrane surfaces, and that's called the intramembrane space, and we're going to see some stuff going on there. Next, we have the matrix, which is kind of like the equivalent of a cytoplasm for mitochondria. It's a fluid-filled space inside of this mitochondria, inside the organelle. The mitochondria also possesses its own DNA and ribosomes. Along with that means enzymes. That's a strong evidence for the fact that this was likely its own organism and leads credence to the idea of endosymbiotic. Now, the main function of the mitochondria is a couple things here. First off, mitochondria can divide, hence that's why we say it's a big deal with endosymbiosis. But also, the mitochondria's membrane has these membrane-bound proteins, enzymes and permeases and ATP synthases. That is very important. So all these little folds here, these undulations, are designed to increase this membrane. Having a highly folded inner membrane is going to give more areas for these surface-bound enzymes and permeases and all that good stuff. Basically, ATP synthase is going to be all over inside this membrane. The more folds you have, the more reaction sites you have. Now, we're going to do this thing called oxidation of pyruvate now, since we made pyruvate. Pyruvate's entering inside the mitochondria, and we're going to go what's often called the link reaction now. The oxidation of pyruvate. So pyruvate is going to enter that matrix. To enter this, it's going to take a 3-step oxidation process. We're going to make some CO2 molecules, a couple of them to be exact. Also, we're going to take NAD positive, and we're going to reduce it by taking, putting an electron on there. We're going to reduce it and also slap a hydrogen on there to kind of make it more balanced or more neutral. We're going to create NADH molecules. So more electron carriers are being made. That's critical here. Also, we're making a special type of coenzyme sugar complex known as acetyl CoA or acetyl coenzyme A. This is what pyruvate becomes. So those two pyruvates are going to become acetyl CoAs, and that is what's going to enter into the Krebs cycle. Now that CO2 that's produced here, it's going to exit the cell through various respiratory processes. It's going to be picked up by the blood cells in mammals, for instance, and it's going to be carried through the hemoglobin and out in the lungs. So you're going to exhale the CO2. Now, plants do some stuff with that. They can often link the CO2 with photosynthesis, and they can do some cool stuff. So once pyruvate oxidizes acetyl CoA, again, this is kind of that link reaction. We're going to get a special 2-carbon sugar known as acetyl CoA, but also we're going to get this NADH and CO2. CO2 is a byproduct. It's going to leave. We're not going to see it again for a while. But the NADH, that's the money maker. This is what we care about. The whole goal here is to make a lot of these NADHs, because those are going to pay dividends later when it comes to ATP synthase. Now, the Krebs cycle, also known as the citric acid cycle, is discovered by Hans Kreb. It took a long time for him to discover this process. It's an 8-step pathway. Each step is catalyzed by a very specific enzyme. Knowledge of the specific enzymes are well beyond the scope of AP biology, according to College Board, and you better thank your lucky stars. Back in, I'm going to sound old here, but back in my day, we had to learn more of the steps. It's not terrible, but it's just another brick in the wall. Back to this though. So it's an 8-step pathway. Each step is going to be catalyzed by very specific enzymes, and because it is a stepwise catabolic process, we're going to end up making a 6-carbon. It's basically a way we're going to build up and break down a 6-carbon citrate molecule. Now, we do have evidence to suggest that it evolved later than glycolysis, and you can see the evolutionary evidence here. So bacteria, for instance, came about 3.5 billion years ago. That's when glycolysis shows up. Oxygen shows up about 2.7 billion years ago, that free oxygen. That means photosynthesis showed up. Eukaryotes, as far as we know, meaning aerobic respiratory organisms, only showed up 1.5 billion years ago. That means the mitochondria came along a little bit later. So what you need to know about this is when pyruvate becomes oxidized during the link reaction, becomes acetyl CoA. Acetyl CoA is going to enter in this Krebs cycle. When it enters into the Krebs cycle, it's going to become citrate, hence the name citric acid cycle. Now, throughout this entire process, it is going to become oxidized. It's an oxidation process. Now, this happens twice. The reason why it happens twice is because you actually have 2 pyruvates for every glucose. So I take one glucose molecule, I split it into 2 pyruvates. Each pyruvate gets a turn through the Krebs cycle. And all while, every time it becomes oxidized, CO2 comes off and it goes as a waste product, much like it did in the link reaction. Now, the electron carriers, again, is where all the action happens. Remember, these are going to pay dividends later. So these are going to be reduced because when you oxidize one thing, reduction of another compound has to take place. The electrons that you remove from the oxidized substance has to go somewhere else. So they're going to be attached to something. And what we see here in the cells is these electron carriers, NADH and also FADH. Ends up becoming FADH2. The graphic doesn't fully display that. So for every glucose molecule, we get 2 pyruvate, get 2 acetyl CoAs, and every acetyl CoA is going to take a trip through the Krebs cycle. And in doing so, we're going to create numerous amounts of NADH. 4 of them to be specific here, and FADH. That's for every turn. And we do this twice. Also, should make a note here, you do make a little bit of ATP in this process. So when we went to look for Krebs cycle, we thought this might have been the big ATP producer. Turns out, Krebs cycle doesn't make that much ATP. So we take glucose and we completely oxidize it down to CO2. We take glucose and we strip it down all the way down to 6 CO2 molecules, and we only get 4 ATP molecules. That's not a lot. Now, when you factor in glycolysis, that's including glycolysis, sorry. When you factor all this together, it's not very efficient. So where's all this ATP coming from? Remember, the main thing the mitochondria does, as pop culture tells you, it's the powerhouse of the cell. So where are all these energy molecules at? Well, that's what comes into the next step. See, these electron carriers. Now, the electron carriers are what the Krebs cycle really does well. The Krebs cycle does not produce that much ATP, but instead it produces like high quantities of these electron carriers like NADH and FADH2. And these are going to go on to the electron transport chain, which is going to be another video. But that is where ATP synthase is done. The work is going to happen right here. They're going to put in some work, and great things are going to happen. Now let's look overall here for the Krebs cycle. For 2 Krebs cycles, so for every glucose molecule, we get about 2 ATP. That's not great. But we do get 8 NADHs and 2 FADH2s. Those are 10 electron carriers in addition to what's already coming from glycolysis. So we're getting 2 from glycolysis. All that's going to come in very, very important. So if only 2 ATP are produced during Krebs cycle, how is that a good adaptation? Well, it helps increase the value of NADH and FADH2. You're producing tremendous amounts of these. So as the graphic there is on the screen, this is like money in the bank. These are going to pay dividends much later into the process. So yes, Krebs cycle itself doesn't make very much ATP. It makes it through substrate level phosphorylation processes. It's okay at it. It's great at this electron carrier action, and that electron carrier action is going to play out really nicely in the next step, the electron transport chain. So let's wrap things up here. So today's video is meant to be a nice little brief overview of glycolysis and the Krebs cycle. The Krebs cycle can be very intimidating if you look at models of it, and I think that did intimidate some of my students. I saw numerous students kind of worried about that. What you really need to worry about is the movement of carbon and really how much ATP and NADH is produced, and also what would happen if I was to disrupt the process, because it is an 8-step process, so any of those 8 steps can be disrupted, and be able to think about what would happen if I prevented it from binding with CoA and becoming acetyl CoA. What would happen if I prevented the citrate from happening? Whatever, prevent it from actually, the NADHs from being produced. Those are the kind of concepts you really need to think about. Don't get so hung up on thinking about all the enzymes that are involved here. This is a very complex reaction. It can be consuming. But in reality, the Krebs cycle is fairly, if we approach it as what it is, it's fairly simplistic, and you'll find that you'll do well with it. So just worry about the accounting aspects and where it happens and some things that could possibly go wrong if you disrupt it. Now in class, we're going to explore this a little bit more. We're actually going to do a respirometer lab where we're going to take and measure the amount of water vapor produced and also calculate the amount of CO2, or at least trying to figure out what CO2 is doing on here. So we're going to do some cool stuff with that. Alright, that's all been today. Thank you all for listening and watching. Hope you all have a excellent day. Thank you.